Okay, so today I think there might be one equation, but that's about it. I'm not going to talk about bioinformatics. It's a bit boring. So what I want you to take away from what I'm going to say today are some of the things which you could do, which would be really useful for all the great skills that you are either acquired or will acquire. And this is applying those skills to the problem of understanding what causes psychiatric disorder, and most importantly, from understanding what causes it, how do we cure it? And so just in case you don't know this, about half of the patients that I would see as a psychiatrist, if they come to me with the most common cause, which is depression, I really can't do anything for. They're really not going to make much of a difference. And those that I can give a treatment to, I wouldn't expect to see any change within about 3 months. And for any psychiatric condition, anything they have, I have no cure. So all we have is symptomatic treatment, and it's not very good. And that's across all psychiatric conditions. And just to put this into context, overall, this is currently the biggest burden on the health system across the world. There is more disability than any other condition. Heart disease, cancer. The psychiatric conditions are now the world's biggest health problem. And we don't deal with this very well. Now you might ask, why is it, given it's such a big problem, that we don't have so much research going into that area? And the answer to that is because until very recently, we didn't really have any tractable approaches. And the basic view of the funding agencies is, what's the point of putting lots of money into an area where we're not going to make any difference? Which is a reasonable point. But over the last 10 years or so, that situation has changed, and it has changed because of genetics. So what I'm going to do in the next half an hour is just run through what we have learned from genetics and focus on what that has told us and hasn't told us, in the sense of how much it will help us understand and treat the diseases that we have to deal with. So why, to start with, would genetics be useful at all? And the answer is, I think, best given by an example from another disorder, from heart disease. It's the same issue, very common disorder, we don't really know what's going on, genetics comes to the rescue, and what we find is that it is possible to find mutations, and this is a very famous gene now, PCSK9, that a group found a mutation in that gene which improved the condition. So we're talking here particularly about a risk factor for heart disease, LDL cholesterol. So if you read what it says here, there's a nonsense mutation, so inactivating mutation that lowers LDL cholesterol, that's bad stuff, going to make you have heart disease, by 40 milligrams per deciliter, and that's associated with an 88% reduction in the incidence of heart disease. That's extraordinary that you could do that. And note, it has to be genetically determined, because if you remove one allele, if you notice here, so if you have just one of the knockout alleles, then it's associated with a 50% reduction. So absolutely what you'd expect in terms of a direct effect from the gene. So if anyone in this room can find a mutation in a gene which reduces the incidence of schizophrenia by even 10%, that would be extraordinary. No one's done that, so that's your task. So I'm going to put this talk into the context of the 3 things I want the genetics to do. So top of my list is I want it to cure the disease. And I've given you one example where genetics could do that. So that's what we're really looking for. But before we get to that, we'd like to improve our ability to make diagnoses. So if you see me, you've got some psychiatric condition, I'll chat with you. That's really what I'm going to do. I'm just going to talk to you. It would be great if we had some diagnostic tests, like other proper doctors, you know, where you can send somebody off for an imaging thing. So it'd be great if genetics could help with that. And then the last thing I've put down is just this, maybe some other stuff we don't know about these conditions. So it's going to tell us interesting new things. So I'm going to take those 3 headings and I'm going to apply them to the things we've learned about the genetics of psychiatric conditions. And I'm going to tell you that we really have learnt only 3 things. We've learnt about the heritability, we've learnt about the genetic correlations, and we've learnt something about genes. So I'm going to go through those three headings. So the first thing is the heritability. So heritability is a measure of the contribution of genetic effects to the disorder. And typically it's been assessed with twin studies where we compare the prevalence in monozygotic, genetically identical, and dizygotic twins. And that's given us, over the last 100 years, a pretty good idea of what the genetic contribution to psychiatric conditions is. There are difficulties with this approach. Most importantly, it's difficult to get large numbers of twins. Secondly, there always is the concern that what makes those twins identical, the monozygotic twins, is not just their genes, but the fact that they're brought up in the same family environment. So there's a confound there that causes some argument about the validity of the studies. In the last few years, we've learnt how to use genetic data, and I'll give you one slide just to try and make this clear. So this is a measure of genetic relatedness that we get from genetic data. So if we look at monozygotic twins, they're genetically identical. Siblings share about half their genetic variation, so they fall in a lump round here. But the data that we now extract heritability information from is here, when we have totally unrelated people. So you'd expect that to be around 0, but it's not quite at 0. There's a spread there. And if you have enough people, you can see some genetic relatedness on the simple assumption that at some point we'll all have an ancestor in common. Think of it that way. So we can use the genotypic data to get heritability. And here's an example of what we get. There's a lot of these now published. These are the conditions. This is autism spectrum disorder, depression, schizophrenia, bipolar, and attention deficit hyperactivity disorder. And the values given up there are less than we've got from twin studies. And there's a lot of argument about why that is, something that concerns a lot of people. So it's about half those, and that's primarily, think of that because we don't interrogate the entire genome, but there are some technical issues. The second thing is it makes less assumptions about the role of the environment, so you don't have the problem with twin studies, so it should be a better estimate. But the important point is the last one. It implies that the psychiatric disorders have a highly polygenic architecture. That's to say, the method wouldn't work at all if we didn't have very large numbers of loci contributing to the risk of disease. So that's what we've learned from heritability estimates. So now I'm going to put that into the three things I want to learn, I want information about, and it doesn't cure disease, it doesn't improve diagnosis, and it's not unexpected. So it doesn't actually help. Useful technique, but doesn't really help. So the second thing we've learned about are genetic correlations, and this is just an extension of the genotype heritability approach. Instead of looking at the heritability of a single condition, we answer the question, to what extent do variants contribute to the presence of more than one condition? So the genetic correlation. And here's some data. So the light green bars are the genetic correlations, and the marks underneath are the pairs. So if you look, and I'm going to talk a bit about this, there's a schizophrenia bipolar, which is marked with a genetic correlation of 0.2. And very recently, about a few weeks ago, this paper came out in Science, and it has a same approach, lots more data. And here's one result that they published, and I want you to concentrate on the schizophrenia to bipolar line. So there's a sort of dark blue. I find this really hard to understand how they lay this out. I wish they had a table. I think they sort of imagine people can't read numbers anymore. But I guess that's about 0.8 on the genetic correlation, and there's a little star in it, which means that's significant. So highly significant, and a correlation of about 0.7 and 0.8. It's from a good group, and it's in Science, so of course we all believe it must be true. But I just want to cast one little doubt upon this, which is, for people who've worked in this field for a long time, they're very familiar with papers which have looked in great detail in family pedigrees at the correlation between genotypes, between diseases. And in short, we don't see in family data the sorts of patterns that this tells us. So I just want to show you, so you're clear about this. Here are two papers from the mid-90s, where two groups have taken a large number of families and they've looked through and they've asked the question, if I have a family member with schizophrenia or bipolar disorder, what's the chance that someone else in that family should have the condition? So remember that the genetic correlation is, from the genotype data, about 0.7 or 0.8. So here's a summary from one of those papers. In personally interviewed relatives of schizophrenic probands, the lifetime risks for all, it says AI, that's affective illness, so that's bipolar disorder. It was, or just bipolar disorder, AI there, was very similar to that found in interviewed relatives of controls. So there's no difference between the relatives of someone who has the disorder and in controls. In other words, there's no increased risk of bipolar disorder if you have a relative with schizophrenia. So that completely contradicts what you'd expect from a genetic correlation of 0.7 or 0.8. And just to make it even clearer, here are some the figures which I pulled out of one of those papers. So the top columns represent the three conditions, the controls, bipolar disorder and schizophrenia, and there's the numbers. And you read this by saying, okay, so if we've got somebody who's got schizophrenia, what is the chance that their child will have schizophrenia? So that's down here. So there's about 5% of the kids would have schizophrenia. And if someone has bipolar disorder, then what is the chance that their kids will have bipolar disorder? And it's about 4%. And if you do it the other way around, because we're looking at the correlation here, someone who has bipolar disorder, what's the chance they have schizophrenia? Well, the children have 0, if you look there, and similarly for the schizophrenia in the other relatives. So it's nowhere like the 50 or 60% that you'd expect from the very high genetic correlation. So this is the first task for you guys. Is this because it's a problem with the maths? Did someone just mess this up? Be really interesting if you could have a look, those of you who like these sorts of challenges, because maybe there's a problem there. And in fact, it came this, I'm showing you this because we had a discussion about this very issue in one of the first of the CGSI meetings, and that led to this paper, which Omar, who was a student then, was attending the conference, and Saharan wrote. So there's definitely stuff you can do here. So just to summarize, genetic correlations, they overturn about 100 years of psychiatric nosology. This is really remarkable change in the way people are thinking. And I'm just pointing out the findings are inconsistent with pedigree data, and it's not clear why at this stage. So now I'm going to score the second finding, genetic correlations, in terms of my 3 headings, and let's see how it does. You won't be too surprised at this, I guess. Doesn't help with disease, doesn't improve diagnosis, but yes, we score 1 for unexpected findings. That's certainly true. So let's move on to the third major area where genetics has made a difference, and that's in the genetic mapping. And by that I mean finding positions on chromosomes that increase the risk of a disorder. I'm not going to go into how this is done, but let's just show you the results. So this has been a very successful enterprise. So this is a genome-wide association study. These are the chromosomes on the bottom here. The likelihood is on the vertical axis, and each of those blobs is a single dot, a single result from a single position in the genome, and the higher the likelihood, the better. And there's this one here, which I heard that Eric Lander called the Freedom Tower of schizophrenia. Like an enormous genetic effect on, relative terms that is, enormous, on chromosome 6. So this has been successful for schizophrenia, for bipolar, depression, cannabis use, and then we're getting into a little more unusual territory, risk-taking behavior. I pulled this off the bioarchive. Intelligence, that's now been mapped. And perhaps most fun of all, insomnia. Look here, there's a gene for napping. Those of you who fall asleep can blame it on your genetic background. There's a gene for napping there. So yes, it's a very successful approach. You can map most things now if you've got a large enough sample size. But I want to bring up a couple of things. So let's go back to schizophrenia. So here's the Freedom Tower. Now, this is a 2018 paper, so there's even more evidence in support of this. And there's about 215 loci. So the first thing is if we just rank all those loci by their effect sizes, you get something like this. So this is the likelihood, the odds ratio, the increase in risk if you have one of those. They're all very small. The Freedom Tower one is the biggest at about 1.3. That's really huge in these contexts. Most of them are tiny down here. And as you may know, even if you add all of these up, you still don't explain more than a small percentage, 10 or 15% of the total heritability of the disorder. So that means, if our assumption about the genetic contribution being polygenic, lots of risk loci is true, that we should keep on finding these for a long way. So it would be really interesting if someone here could work out how many. So we're up to about 250 here, and let's suppose that it's 1,000. If it's 1,000, that's already, if it's one locus per gene, which I think is a reasonable assumption, that's 1 in 20 genes, but that would only be 4 times what that is, and these incremental increases are very small, so it's probably not going to be 1,000, 2,000, 5,000, suppose it's 10,000. That will be half the genes in the genome. Are we really prepared to believe this? So there's a sort of unanswered issue around how many genes this approach really implies, which is definitely worth thinking hard about. And the second thing I want to show you is a bit of unpublished data. You can pull this off the internet, which is mapping genetic effects for schizophrenia in East Asia. So very successful, we've got lots of peaks, but something missing? No Freedom Tower? Why is that? Let's just make this clear. There is something on chromosome 6, but it's not the same. So here's the European one, and here's the East Asia one, so it should be there. There really isn't a hint of it. So if people start claiming that the biology is the same between different parts of the world, you can always point to this example and say, no, Dr. Flint tells me it's not true, because it's not true. That locus is missing. So there's an interesting issue there. So just to summarize, genetic mapping of any psychiatric disease is possible. I think that should be pretty clear to you. It identifies small effect loci consistent with a major polygenic architecture for all psychiatric diseases, and I've argued that that does cause a few upsets. So let's put that into my 3 categories. Doesn't help to cure disease, doesn't improve diagnosis, and no unexpected findings. Okay, last thing. So this is really where we should be getting down to business. So this is, we want to find the genes because these really might help us understand what causes the condition, and we might find cures. So let's deal with the first thing. The first thing is that we have lots and lots of loci, 250 for schizophrenia and so on. All of those, we now know, are genetic effects that lie in regulatory regions of the genome. They are not genes. And here's one example to make that clear to you. So this is not for schizophrenia, but the argument is the same. This is for platelet count. So there's a locus there on chromosome 9. You can see in red it says RS something something. And then there's a big arrow when it says JAK2. So JAK2 is the gene, and this is the variant that causes a change in platelet count. And that little green mountain there, that's a regulatory region marked by, in this case, a DNA's cleavage site. So it's where proteins are binding to the genome and doing stuff. And it's regulating the expression of that gene. So this is typical. We've found many instances of this now, where a variant alters the regulation of a gene, which is quite a long way away. So you might think that that locus there is responsible for change in RCL11 or MIR101.2, or it's not. None of those. It's actually regulating that. So bear that example in mind when anyone tells you that from a GWAS they know the genes. They don't. They just know the positions of risk loci. However, I have to say that doesn't stop our colleagues publishing papers like this. Okay, that's not true. But we all do it. And one reason that people do this is because they then take all of those gene names, which they assume are associated with those loci, and they ask, what do they have in common? And it's very common to find papers with lists like this, where they've taken all of these This is for bipolar disorder, schizophrenia and depression. They take the loci, they find the genes, and then they ask what do they have in common, and they come up with some pathway analysis, some annotation in a database that tells you what the gene does. And I think there's one important point just to take away from this. So you don't need to be a neuroscientist to realize that if I say that schizophrenia is due to genes which are involved in the neuron, that's the postsynaptic areas, the axon, is not a deep insight. Like if I were to tell you schizophrenia is due to genes that act in the little finger, you go, wow, that's really interesting. But in the brain, in the neuron, not so insightful really. So you might argue, okay, well it says histone H3K4 methylation and dendritic spine. I mean, come on guys. This isn't telling us anything really fundamental about what causes the disease. Now this may be because it hasn't been looked at by great bioinformaticians who can pull information like this and turn it into a real hypothesis about what schizophrenia is. Maybe that's what you should be doing. But I suggest you treat these with a bit of caution. There is one approach we can take which does get a lot further along the path that I want to go, which is to take one of the genes and really work out what it does. And there's one paper, and only one paper in schizophrenia that does that. This is a paper by Steve McCarroll. It was published a couple of years ago in Nature, and he worked out that the locus, which is on chromosome 6, part of the freedom locus, actually affects a complement component, not an HLA gene, which is where most of the other loci, most of the other genes are at that locus. And he comes up with some hypothesis, which I won't go into, as to what that gene does in schizophrenia. That's really hard work, and Steve deserves getting a nice Nature article out of that, because it was a lot of work and unexpected to get that far. That's one gene. Another 249 to go. It's a long path. So taking the GWAS hits and turning them into mechanism is going to be difficult, and we definitely need to think hard about how to do it. The second way of finding genes, though, is turned out to be a little more direct. Now, the example I gave you at the very beginning was a PCSK9, and what the group there had done was sequenced the exomes of lots of people with a disorder and looked for mutations. And you might think that would work in schizophrenia, but there's a good reason why it shouldn't work, and the good reason why it shouldn't work is because we've never ever documented a case where schizophrenia is due to a mutation in a single gene. Never. Lots of looking for this, I have to tell you, lots and lots of looking. Not schizophrenia, not bipolar disorder, not major depression. So it was a great surprise when a couple of groups reported this. This is for autism and this is for schizophrenia. And what they've done is they've sequenced the exomes of a small number of patients, and what they're reporting is that there is an increased number of mutations in the exomes of people with schizophrenia compared to controls. So this was hugely important because it meant you could go straight from those genes, straight from those mutations, I mean, to the gene and be certain you'd found the gene. Except all they're actually reporting is an increased rate. So it's like I count up all of the mutations in people with schizophrenia, they got 50. Count up all the mutations in those who don't, and they got 30, and that's significant. It's not telling me what the genes are, but it's an indication if I could look in more detail, I should find out what those genes are. And it's generated a lot of interest. These are all germline mutations, yeah. It's very straightforward, nice, easy experiment. But the problem is it hasn't really helped us because it turned out, which you could have guessed, I think, from my comments about the polygenic architecture, that there are so many genes that are involved in this condition that in order to find multiple mutations in a single gene, which is what you'd need to find to show that the gene is causative, you need to sequence not hundreds, not thousands, but probably tens of thousands of cases. And that's not stopping people doing it, so that's ongoing at the moment, and those results will come out. But it's proved to be a lot harder. There is one counterexample, which is in the case of autism, where this approach has worked. And with autism, people have sequenced and have found genes. I'll give you one example. I think this is a useful one, which is a gene called CHD8. And this is published from Evan Eichler's group about 4 or 5 years ago. And what I'm showing you here, it's a slightly unexpected picture maybe, and the vertical axis is the head circumference of kids, and this is their age. And what he's showing here in the red line are those with cases of autism who have the CHD8 mutation. Now, it had turned out that in the literature there's been a long story about kids with autism having big heads. Really odd. Like, there's also stories about them having poor digestion and constipation, all sorts of strange things, which most clinicians, they note, but they don't take much interest in, because we can't explain it. It's part of the syndrome. What this discovery showed is that actually those clinical features do point to the specific effects of mutations, because the people with CHD8, that mutation appears not only to affect the social behavior, the intelligence of the child, the language capacity, but also the head size. So that was intriguing. And that points to a couple of things I wanted to be able to do, now I now have a test. I now have a biological test. If I have someone with autism, I can send them for screening for this mutation, and that gives me some useful information. So that's really important. And we'd like to see that happen again and again with other conditions, but we're not there yet. So exome sequencing is the second way of finding genes. Can get there, but hasn't yet. And there are some fundamental problems with the screening issues and so on, which we don't need to go into, but it's potentially possible. Yeah.
Just to fill gaps in my background. But how inborn is, I can imagine autism is very inborn, like the child. So how the life history? Because I guess
autism is probably one of the most genetically determined conditions.
But with schizophrenia, I was wondering.
Schizophrenia is about 80%.
Okay.
Yeah. They're all pretty heritable.
Mm-hmm. What is pretty heritable like?
The condition.
No, yeah, but what do you mean by pretty heritable?
Pretty heritable, more than 40%. Will that do as a definition?
Oh, yeah. Just wanted to...
I think 5% is not very heritable. Put it that way. It's conventional. So I want to move on to the third way. And the third way that people have found genes is by looking at deletions in the genome, bits missing and bits added. And schizophrenia has been the classic case. Okay, don't know how to get rid of that. Shouldn't obscure the diagram. If you look at the one at the bottom here. So what we're looking at is this is a normal patient, and this is someone with schizophrenia. And the experiment here is to label a piece of chromosome 22, and that's the green here. So you can see that there's 2 green and 2 green in that one, and label also a small region towards the middle of chromosome 22 with this red probe, and you can see there are 2 dots there, but only 1 in that screen there. In other words, there's a piece of DNA missing, and this comes by different names. Structural variants, copy number variants, copy number polymorphisms. You'll see various descriptions of this with vague justifications for what they are. But the important point is, because it's a small piece missing or added, we can look at what that piece of DNA contains, and just as we did with mutations, we can say, okay, this must be the cause. So that piece of DNA missing, taken out that gene, therefore I can find a gene. So it's another way of getting at genes. And here, this is from Jonathan Seabat from last year. If you look at the top, this is now looking across the genome, not just you'll see chromosome 22 lights up. There are a number of places in the genome where he finds evidence for a deletion at the top, and at the bottom here there's an increase, and 16p is well known as a risk locker. So there are places in the genome where if you change the amount of DNA, you will find a change in the risk. But unfortunately, often quite a lot of DNA is missing. So the chromosome 22 locus is about 2 megabases, and on average, a megabase contains about 10 genes or so. So there's about 30 or 40 genes. And it's really hard to know which is involved. So we still have a problem taking a finding like that and turning it into genes. Okay, so I'm almost at the end. So I've told you about heritability, I've told you about correlations, and I've told you about mapping, and I've told you about gene finding. And I've said we are finding genes now, but we're having a real problem with understanding what those genes do in relationship to schizophrenia. And I've pointed out that the literature is full of figures like this, or this, where people have tried to understand the relationship between those genes and create some pattern. But I have to say, as a biologist or even as a doctor, this is garbage. I mean, come on. What is this telling us? It really needs some good bioinformaticians to turn this into something meaningful, because I can't use this in the clinic. I can't hold that up and say, your son has got one of these. It just doesn't work, you know? No, we need something a little bit more, caring. Okay, so now let's go to the very last slide, and now I'm going to summarize everything that we've said. So for the genes, we're not getting to curing diseases, but we're going to improve diagnosis. I think there's a little hope there. And unexpected findings, yeah, there's quite a few of those going on, no doubt about it. But the top line is the one I'm most interested in. So that's your job, okay? We've generated all of these data. Please go ahead and work out how we can turn that really interesting genetic information into a cure for the world's commonest causes of disability. What are you waiting for? Yeah.
I know that you focus on genetics, but what about genetics? Or do you choose the diseases which are primarily inborn, or as you said, heritable, which don't need
Okay. So maybe I should have explained this. So the first thing is that all diseases are heritable, really. In fact, everything's heritable at some level. Even what color car you choose, and they all have some heritable component. The reason that that's useful to us is because when you know nothing about the cause of a disorder, when people have worked on a condition like schizophrenia for decades, for centuries, and still don't know what causes it, you can take that genetic component, which might be small, maybe 10, 20% sometimes, and you can run it through these genetic analyses, and you'll map it, and you'll find genes. So it's really important that we have these techniques to push us forward. But the thing is, what to do next?
So, can you give us a clue what can we do next?
So the first thing you can do is, I need some clever mathematicians to go through all of those stupid heritability and correlation calculations and just work out what's going on, please. That would be really, really helpful.
So Jonathan, have they looked at large pedigree data sets where they don't observe correlation?
So they don't have, no one's got their hands on data sets where they've got, so the places where there's no correlation are sort of hand-curated, interviewed patients, all done in the 90s and so on. The only one I know where they've got DNA from is the one from, there's a big study that, in fact, Ken Kendler did in Ireland, so they have 300 families and they do have DNA from those. But I think the other ones are all, of course, because at the time these studies were done in the 80s and 90s, no one thought it would be possible to do what we can do now, so they never took the DNA.
So you're saying you suspect that there may be a problem with the whole heritability approach?
There is a problem with the whole heritability approach.
Okay.
But what I don't understand is, you just said even if you know the disease, so what can you do to fill out the problem?
You work on the genes. So you try and take one of those sort of furball things with little spikes, and you try and make that into something sensible that I can use as a biologist to advance my understanding of the cause of schizophrenia, autism, whatever.
So can you give us a little insight onto when Steve McCarroll found that it was actually that gene? How did he do it? Yeah.
So that's so the way he does that is takes the approach that I showed you briefly in terms of saying, here's the locus. So I have a slide where I showed you there was a locus influencing platelet count. And the first question is, what is that locus doing? And it's involved in a regulatory element in DNA. I can prove that molecularly. I now want to know, what does that regulatory element do? And again, I can carry out an experiment which shows me which gene that regulatory element regulates. So that in brief is how it's done. But it's a lot of hard molecular biology.
So in terms of using the data in order to cure disease,
Yeah.
so it sounds like if you have a hypothesis about a specific gene, then we need more experiments. But how can you utilize the current data to...
Oh, that was the question I was putting to you. I don't know. If I knew the answer to that, I'd be doing it.
Okay, so no one is doing...
Oh, they're trying to, but they're not very clever. They're not as clever as you guys. They need help, you know. Of course people are trying, but it's a hard problem. But it needs, like, you know, people coming in from a new angle, new thoughts about this. I think, you know.
Was it done in other diseases that were successful that we can kind of copy or try?
Okay, so as a general rule, all diseases that are approached in this way, all complex diseases, have this problem. So it's not that there's very much that we could learn. The things that are useful is that the exome sequencing will give you things like the PCSK9. So people have looked hard and found other cases. So I didn't give you this example, but in diabetes, there are now mutations known that do alter the risk. So that's just a brute force approach. Let's just take thousands and thousands and thousands of people with schizophrenia or whatever and see if we can work out. A more thoughtful approach, there's a paper by Aravinda Chakravarti, he doesn't work in this area, but he's a good geneticist. He says, look, let's take people who we think, for various reasons, will have a much increased genetic risk for autism. So it so happens, for example, that women don't have, there's a lower prevalence in women. And if you have family relatives, is that going to increase or decrease your risk? You can sort of work out where you'd expect to see a genetic risk. And if you sequence those people, then you have a much higher chance of finding penetrant stuff, and Aravinda has shown that. And another approach might be to think of this in terms of, someone raised this, about somatic mutations. Are there cases where there's independent mutations arising, maybe different parts of the brain? There are a whole series of things that one could apply, just thinking hard about how genetics might operate. But I came to you guys mostly because I know that you deal with large data sets and you deal with annotations, and those sorts of pictures I showed you are really, at the moment, all that those approaches will generate. And I think we could do better, but I don't know how. I'm sorry. You had a question.
Well, I was going to ask what your thoughts are on the Pritchard omnigenetic model. Like, is that getting at why you can't boil this down to? Is Jonathan right in some sense that like all genes that are expressed in the tissue of interest are involved?
So your question is going back to the question I raised, which is if you look at the tail of the genetic effects, and does it go to infinity, which is what the polygenic model or the Fisherian model implies? Yes, that was your question. I don't know the answer to that. And I'm not that happy with the idea. So there's an idea out there, for those of you who don't know, that you can deal with this problem that you've got so many loci being involved by saying that there might be a core set, that there are some which are sort of peripheral and some which are core. But the problem with that argument is, and I gave you the evidence against this, is that if you take what looks really important when we know the mechanism from Steve McCarroll, this chromosome 6 locus in Europeans in schizophrenia, doesn't work in East Asians. So is this a core mechanism or not? I mean, this is difficult. We don't know the answer to that.
What is that? Is this a power issue or? Because, when you have more mutations in the exome, could be explained by the study.
So epistasis means the pairwise or higher order interaction. So you can have, if you've got, the simple way of thinking about all of these conditions is there might be a choice of 1,000 loci that are predisposing. And if you have 100 of those, any 100, it increases your risk above a threshold and you're ill. That's a simple way of thinking about it. But it may be that the contribution is not equal. So if you have locus 15 and 25, rather than it just adding up, they could multiply. So you could have an interaction which isn't additive. And is that going to explain some of this? I don't know. I'm sorry, I'm not doing very well at the questions. Yeah.
Is there a ratio of, I'm not sure which disorder you've presented, European versus Asians? Is there a different ratio, different prevalence?
Different prevalence of
that specific disorder that you presented in Asians?
Do you mean is schizophrenia more common in... No, it's the same. Yes. It's pretty much the same everywhere. It's one of the reasons why people have thought it's primarily a genetic condition. I mean, there's some variations, but it's not marked. It's about 1% on average, with fluctuations 0.5, maybe down to that level. Depends on it's a little difficult to get this clearly because the diagnosis is not that straightforward on large enough numbers. So if you want to get a real accurate estimate, you're going to have to interview lots of people, and that's expensive and difficult. But roughly, it still holds. It's pretty prevalent in all places. Okay.